Ура! Сегодня я прямо хорошая девочка. Спасибо большое. Good morning, everyone. Guys, start collecting to listen to a lecture on Mycobacteria tuberculosis. Guys, start collecting. Do you hear me? Good morning, guys. Start collecting. Lecture starts already. Guys, where are you? Ui. Guys, students, first division, start coming. Oh, good morning, everyone. My dear, you are the first one. Good morning, guys. So, today we'll discuss Mycobacteria tuberculosis. And I would like to remind you that the last lecture had been devoted to very dangerous bacteria that cause quite rare but dangerous diseases: anthrax, cholera, and plague. And I'm not sure whether you will see so many cases of that three infections, but I'm absolutely sure that you will see in your medical practice a lot of cases caused by Mycobacteria tuberculosis, because this is the one of deadliest and commonest infection in the world. And at the very first slide of our lecture, you can see how macrophage engulf Mycobacteria. See, this is a very nice picture, and try to digest them. But as far as we know about the ability of Mycobacteria to escape immune response, so usually it is very hard to eliminate it from the body. And today we'll discuss this feature and many other features of Mycobacteria. So we go ahead with a bit of history, because you have to know it, that Robert Koch was the one who identified Mycobacterium tuberculosis as a causative agent of the disease. So, because he managed to develop approach to stain this organism and to culture it. Before works of Robert Koch, they thought that tuberculosis was inherited disease. But then it was Robert Koch who proved the mechanism of transmission of Mycobacteria, identified the microorganism as a causative agent of the disease, and demonstrate the same disease in laboratory animals. He also developed tuberculin, but he hoped it was a remedy or for treatment or vaccine against tuberculosis. And due to incorrect use of tuberculin, several patients had been infected and died from the disease. After that, Robert Koch was really disappointed and upset, and he left for Africa and he stopped working with the Mycobacteria tuberculosis and continued his work with different infections. But the works of Robert Koch was very difficult to overestimate, and in 1905 he was awarded the Nobel Prize for his research work of tuberculosis. So what By the way, Mycobacteria is a very large group of bacteria. You can see here that we may see there are more than 100 species, but they are mostly saprophytic. We have a lot of Mycobacteria that reside in animals, birds, and others. So also they may reside in the nature, so they can be saprophytic and parasitic for other hosts. So they are usually, regarding the morphology, rod-shaped organism. You can see they are tiny, fragile, long rods. And usually they are strictly aerobic, especially Mycobacteria tuberculosis itself. And the causative agent of TB is strictly aerobic. Then one more thing that distinguishes Mycobacteria from other organisms. It has a very unique structure of the cell wall. And usually bacteria of that features of the cell wall are called acid-fast bacilli. So a bit later on when we discuss the structure of the cell wall, you will understand what does it mean. And also a lot of Mycobacteria are slow-growing organisms, not all, but the ability to grow very slow can be very important cultural differential characteristics to identify Mycobacteria tuberculosis itself and to distinguish with some not so slow growing Mycobacteria. And one more common thing for Mycobacteria, they don't form spores. So, as we said, in the group of Mycobacteria, we have saprophytic, pathogenic, parasitic with different potential of pathogenicity. So the primary or frank pathogens that belong to this group are at least two: Mycobacteria tuberculosis, and then we'll collect some more species in so-called TB complex. And Mycobacteria lepra that causes leprosy. And here you can see an example how the case of leprosy looks like. We'll mention it one more time. Then second group of Mycobacteria are so-called atypical Mycobacteria. The good example of it is Mycobacterium from avium intracellulare complex, MAC complex. Mycobacterium avium intracellulare complex. Avium, it means birds. So these Mycobacteria don't infect usually people with normal immunity, but only those who are immunocompromised. And because now we have plenty of immunocompromised people due to HIV infections, so most probable target for this complex Mycobacteria are patients with AIDS or HIV infected. But also occasionally, quite rare, but they may cause diseases in persons with generally normal immunity, but that's extremely rare. So, because the whole lecture will be devoted to Mycobacteria tuberculosis, I would like to say just few words about Mycobacterium lepra, the causative agent, and leprosy. In leprosy, the nerves and skins are principal sites of disease. And that's why it's usually result in deformities and lesions. Leprosy may occur in two general forms. So regarding the ability of the body to develop immunity. One type is lepromatous leprosy, and we call it energic disease when body fail to develop immunity, protective immunity against these microorganisms. And people with low immunity develop the very huge bacterial load, and it is very serious disease. You can see the examples of deformations and lesions in this disease. And the second type is so-called tuberculoid leprosy, and these occur when the body is hyperreactive against Mycobacteria lepra antigens, and because of the altered, exaggerated immune response, the body will form excessive granulomas, but because the immunity is working, and that is the example of tuberculoid leprosy, the viral load or the bacterial load in this disease is very low. We won't discuss leprosy anymore, it's not within our task today, so we concentrate on mycobacteria. So if we mention the species, so all species that may cause TB are placed into Mycobacteria tuberculosis complex. And the reservoir are humans, or in case of Mycobacteria bovis, animals. And disease cost is called tuberculosis, and it causes millions of cases annually. Leprosy is also quite common, but it is endemic mainly in Asia, Africa, and Latin America. So, first, the name of Mycobacterium tuberculosis. Myco means fungus bacterium, so somewhere in between. And that originate from the ability of mycobacteria to form thread-like structures, and once it had been confused with fungal infections. So when we describe morphology, is a very typical bacilli, thin, slender, straight rod. So up to 3 to 5 micrometers in size. It is polymorphic, it means it may change its shape from coccoid to filamentous forms. So mycobacteria can be stained, but our regular gram stain is not in use because with the help of gram stain, we cannot differentiate mycobacteria with other bacteria. So this is why this special type of staining is used. Quite often we may use fluorescent staining. I would like to underline, not immunofluorescent staining we mean right now, but fluorescent staining. Why it is useful for TB identification? Because the organism has predisposition to be stained by yellow orange, auramine, or rhodamine, but not the other bacteria. So because it is enriched in lipids, the cell wall is enriched in lipids, then the fluorescent dye dyes the mycobacteria much better and binds with mycobacteria much better than with other tissues or bacteria. So that's why we may use direct fluorescent staining. But the classical example of staining is the one of the modifications of so-called acid-fast staining that is called Ziehl-Neelsen staining. It is used in several countries as the primary staining for mycobacteria, but then in some other parts of the world, other modifications of so-called acid-fast staining may be used also. So we use this and then we discuss how to perform it. So, Zell-Nielsen staining depends on the structure of the cell wall because it is called acid-fast staining. What does it mean, acid-fast? It means the 95% ethyl alcohol containing 3% of hydrochloric acid will not decolorize acid-fast bacilli, while it easily decolorize all other bacteria within 30 seconds. So generally acid fastness is the ability to the cell wall of this type of bacteria to stand decolorization with acid alcohol within 30 seconds. Acid fastness depends very much on the high quantity of oxygen lipids in the cell wall of Mycobacteria, tuberculosis, and a lot of other Mycobacteria. That's about the appearance under the microscope and staining. Now, cultural characteristics. So we can culture Mycobacteria in non-selective media. For example, if we take some specimen from the blood or cerebrospinal fluid in case of generalized and atypical TB. But most probably we use sputum, so that's why most commonly selective media are used for primary isolation of Mycobacteria. So, Mycobacteria are very fastidious organisms. To grow in the laboratory, it needs a lot of growth factor that we will mention them, but generally say we may have several types of media that can be used for the growth. The most common, of course, is selective medium, as I said, because the most common specimen taken is sputum. Usually selective media contain antimicrobial substances like antibiotics or disinfectants, for example, malachite green. So then it gives a very nice color to a medium, malachite green. See, that is the color of most common medium. So the adding of the antimicrobial substances is very important because, as I said, Mycobacteria tuberculosis is the very slow growing bacteria. So if the contaminants are not in the growth of contaminants is not inhibited, so they overgrow this microorganism very easy. So we won't see it anywhere. So three general formulations are used: semi-synthetic agar medium, inspissated egg media, and broth media. We will concentrate on inspissated egg media because this first is not very sensitive, less sensitive to the growth of Mycobacteria. The best medium recommended by WHO for primary isolation of Mycobacteria is so-called Lowenstein-Jensen medium. See what does it contain: salts, glycerol, malachite green, see the very nice color, and complex of organic substances. I mean, Mycobacteria, they like what we like: fresh eggs, egg yolk, potato flour. All those components are growth factors for Mycobacteria. So, and antibiotics and malachite green make this medium highly selective. And this medium is very sensitive for the growth of Mycobacteria, and very small inocular in specimen is needed. The inoculum of the medium yields grows within 3 to 6 weeks. My dear, still they grow very slow, so that's why time is needed to get the growth of Mycobacteria. So this is very important that bacteria grow better in broth medium. So in fact, it is true for any type of bacteria, including mycobacteria. But for mycobacteria tuberculosis, it is really critical because we can get the growth and identify the disease much faster than if we would use selective um, uh, solid medium. Okay, so usually we add into this medium enriched, it is enriched with bovine serum albumin. Then also we add twins, and this is water soluble detergents. Why? Because mycobacteria, because of the lipophilic surface composition of the surface structures, grow in clumps, and they also have the tendency to form the masses. And those masses, take a look. This is called, for example, serpentine cord. It looks like a snake. But the snake is made up of individual mycobacteria organisms. So this is because surface is highly hydrophobic. Twins is added to disperse bacteria growth better. So they will be better recognized. Broth medium is used usually in automated culture systems. You can see the automated culture system back there. So that's quite new, and usually the growth in this medium is more rapid, and small inocula is needed for proliferation of bacteria. So the growth can be gotten within 5-7 days. What are the cultural characteristics of Mycobacteria tuberculosis? So as we said, they are obligate aerobes. Increased carbon dioxide tension will enhance the growth of the capnophilic bacteria. Biochemical activities are not characteristic, and this is for two reasons. First, they are varying, and second, we should grow wait too long until we can check the biochemical activities. So they are not really in use in identification of bacteria. This is some of the examples when biochemical tests are not in common use for identification. So the generation time of this bacteria is much higher than for others. So, and the generation time, you studied it in general microbiology, is about 18 to 20 hours. Compare with E. coli generation time, that is 20 minutes. Of course, they will overgrow mycobacteria if they are placed on the same medium. So, Mycobacteria tuberculosis considered to be more resistant to some chemicals due to their hydrophobic nature of the cell wall. They also may resist some antibacterial agents, for example, malachite green, some antibiotics like penicillin. Then they resist acids and alkalis, which are used to eliminate contaminating organisms from the specimen, and this approach is also used to concentrate mycobacteria in the clinical specimen. So the drying also won't kill mycobacteria very quickly. They survive for very long time in dried sputum for half a year up to a year. Mycobacteria survive in milk and other organic materials and on pasture land so long as they are not exposed to the ultraviolet light. That is not good for mycobacteria because it is sensitive to UV light, temperature. If you are going to pasteurize milk, so then it will kill mycobacteria. And also they are not so resistant to alcohol and formaldehyde. So now a few words about the virulence factors of bacteria. Interestingly, that, oh sorry, interestingly, that uh, bacteria that cause very severe disease, but they are not really toxigenic. I mean, they don't produce typical exotoxins. And most probably pathogenicity and virulence factors or virulence of mycobacteria is attributed rather to the chemical structure of the cell wall than to toxigenicity. So first, a very important part, plenty of different lipids are largely bound to proteins and polysaccharides and incorporated into the cell wall. And there we have mycolic acids. You can see the simplified structure of the mycolic acids. Then also phosphatids, waxes, and cord factor. All those lipids are responsible for the granulomas formation during the disease. They also, phosphatides, for example, are responsible for caseous necrosis. And altogether, lipids, waxes, and proteins are responsible for hypersensitivity reaction. Proteins, when bound with the waxes, can induce tuberculin sensitivity that we check in MAN2 test that we discuss a bit later on. Then the backbone polysaccharide can induce the immediate type of hypersensitivity, mainly of type 1. So you can see here better the structure of the mycolic acid. So you can see the arabinogalactan macrolate layer bound with the peptidoglycan layer with the glycans and acetyl glucosamine and acetylmuramic acid cross-linked with the peptides. Then you can see a lot of polypeptides incorporated into the cell wall. And also you can see not connected with mycolic acids, free so-called lipids, waxes, mycosites, and cord factor. And here you see beneath the cell wall, we have a cytoplasmic membrane. Here is a very good picture to compare the structure of mycobacteria cell wall. You can see them here with the cell wall of other organisms that we have studied already. These are gram-negative bacteria C and with its lipopolysaccharide. Gram-positive bacteria with their teichoic acids. Even fungi, do you see, with a lot of mannose binding bound proteins in the cell wall, followed by beta-glucan and chitin lipoproteins in the cell membrane. And finally, you can see here mycobacteria. So that the part that is exposed to the environment, I mean for our body, are the mycolic acids with the glycolipids on top, and then arabinogalactans bound to the mycolic acids, then thin layer of the peptidoglycan, then the cell membrane. Now, a few words about the pathogenicity of lipids. Mycolic acids are long-chain fatty acids, and they complex with moramyl dipeptide and cause granuloma formation that we see in histology. And phosphatides induce the so-called caseous necrosis, which is named because of the cheesy appearance of the tissues in the granuloma underwent caseous necrosis. Voxes are also involved in hypersensitivity formation, and the major factor that causes this serpentine cord formation. Do you remember we've discussed it already? This is so-called cord factor, trehalose dimicholate, that is responsible for it. Here, mycobacteria are arranged in the parallel chains. And apart from the morphological feature that depends on the cord factor, the cord factor also may inhibit the migration of leukocytes towards the site of inflammation. That also may cause chronic granulomas formation, activating the immune system, and also can serve as immunological adjuvant. And we have this type of adjuvant that is called Freund adjuvant. But we cannot use it for humans because usually we use it in research work as adjuvant to vaccines because it causes too strong inflammatory response in the body. Now let's take a look at the distribution of Mycobacteria tuberculosis cases over the world. And my dears, this is 2018, and that is the latest global statistics available right now. You should understand the WHO meets a lot of difficulties in collecting the statistic information from the whole world. And the last 2 years also, it was difficult within the last 2 years because of the coronavirus infections make a mess out of the healthcare facilities in a lot of countries. So that's why even with this we have some delay in the collecting of the epidemiological data. But you can see here that in Russian Federation, where you live right now, the incidence per 100,000 population per year is about, is it, I think, 100 to 199, right? So let's take a look at India and let's take a look at Africa and some parts of South Africa, right? So here the incidence is so high. So in India it's around 200 up to somewhere 500 cases per 100,000 per year. So this is the worldwide spreading of the mycobacteria. So a few words more about the global statistics. Altogether, we believe 1/3 of the world's human population has been infected with Mycobacteria TB, that cause TB. About 100 million individuals are infected annually. About 10 million individuals develop overt disease annually. And each year, 4 to 5 million become open or infectious. I hope you know what does it mean, open cases. This is when the patient excrete bacilli with the sputum by coughing. So that is really dangerous because this is the direct way for infection that may spread quite quickly. And moreover, each year around 1 to 2 million die out of mycobacteria infection. And because we have a lot of students from India, I would like to underline that India is the highest TB burden country, accounting for more than 1/5 of the global incidence. Take a look here. So, global incidence around 9.4 to 10 million, and Indian dual incidence is around 2 million. So that is really very dangerous. So, and you can see the input of other countries there with the high incidence of the disease. China, South Africa, Nigeria, Indonesia, and so on, right? Let's take a look at the pathogenicity of Mycobacteria tuberculosis. Uh, mm, Not only humans, but also guinea pigs and some other animals are highly susceptible to Mycobacteria tuberculosis infection. Cattle is resistant, but then the cattle has their own mycobacteria that is called Mycobacteria bovis. And interestingly, that humans are equally susceptible to both Mycobacteria tuberculosis and Mycobacterium bovis. The route of infection determines the pattern of lesions. And we have two routes, respiratory or intestinal. Most typical, of course, respiratory. Some atypical mycobacteria like Mycobacteria kansasi may produce human disease that can be indistinguishable from tuberculosis, but they are not so common. So other types of mycobacteria may cause only opportunistic infections and usually don't enter deeper into the body to the bloodstream and may cause the only surface lesions. So the variety is high. Generally, what is TB? It is a long-term infection caused by acid-fast mycobacteria that belong to TB complex. What do we consider as TB complex? Not so far ago, Mycobacteria microti was added, and together with M. tuberculosis bovis and africanum, they make so-called TB complex. Exposure is by breathing or eating infected droplets. And so these air droplets will reach lungs and affect lungs. And from the lungs, they may be spread within the body and cause mycobacteria lesions or infection of almost any organ of the muscles, of the bones, of the internal organs like kidney and so on. During the disease, mycobacteria reside intracellularly in monocytes, reticular endothelial cells, and giant cells. My dears, these mycobacteria are called facultative intracellular parasites. Why? Because they may grow outside the cell. They don't need it really for growing, but if it is ingested by monocytes, macrophages, and reside inside the cell, they may also grow there. For mycobacteria, this intracellular location favors microbial persistence, and for us, this intracellular location uh, makes chemotherapy not so effective because it is difficult to reach microorganisms when they reside inside the cell. Intracellular location protects them very much. Resistance and hypersensitivity of the host greatly attributed to the development of the disease. So, being not so highly toxigenic and unable to produce exotoxin, mycobacteria cause still very serious disease. Within the TB, there are two principal types of lesions that can be formed in a body, exudative and productive. Exudative type is acute inflammatory response, and it is characteristic of pulmonary disease. The lung tissues resembles bacterial pneumonia. In lung tissues, it resembles typical pneumonia, and it accounts for the edema, accumulation of the fluid, the edema fluid, polymorphonucleated leukocytes infiltration, and monocytes surround bacteria, mycobacteria. During this phase, about 6 to 8 weeks after initial infection, the tuberculin test start to be positive, so that explains the hypersensitivity, development of hypersensitivity reaction. What can be the outcome of this type of exudative type of the infection? The spontaneous resolution, maybe yes, it is possible. Then the development of necrosis of tissues as a result of phosphatids and other virulence factors of mycobacteria. And most probably we have progression and development of the productive type of lesions. So they are called tubercles or granuloma because as the formation of these tubercles is the result of body immunity response towards mycobacteria. So this is a sort of chronic granuloma, and it consists of 3 zones that you will study in pathomorphology, but then anyway you need to understand what kind of lesions are they and where do we have microbes there finally. So the microbes are in the central area of the lesion, and usually they are in large multinucleated cells that are called giant cells, and mycobacteria are here, right? So then we have a mid-zone of so-called pale epithelioid cells. What kind of cells are they? Not epithelial, no, my dear. These are activated macrophages that are ready to perform oxygenic bursts to kill mycobacteria. And then in the peripheral zone, we can see fibroblasts and a lot of T lymphocytes, T helper, and to less extent T cytotoxic lymphocytes, and not so many but monocytes. So the later on, peripheral fibrous tissues develop, and the central area undergoes caseous necrosis because still mycobacteria are affecting macrophages outside. So the possible consequences of this is the cavity formation, because the caseous necrosis, and then the microbes break into bronchi and empty is constant there, or then fibrosis and calcification of the lesion. You can see different types of granuloma, caseous granuloma with the necrosis inside, then non-necrotizing granuloma, and fibrotic granuloma, but we have some common property in all of them. See, inside granuloma, we have alive Mycobacteria tuberculosis, even in fibrotic granuloma. What does it mean? It means even if our immunity suppresses the growth of microorganisms, they are there still alive. So, and while they are still alive, we always may have some possibility for the mycobacteria to start growing again. That's why we need to monitor all cases, even resolved cases of Mycobacteria tuberculosis infection. And here also good, you can see what main type of cells participate in the granulomas formation. See, comparatively few neutrophils, right? Mainly macrophages, right? And then apart from them, we may see B cells, CD8 positive lymphocytes, E and T helper cells, and covered with fibroblasts quite often. So, and here you can see the comparing. So, because the chest X-ray examination is the very common pre-laboratory method for diagnosis, so you will learn how to read it. And in advanced case of pulmonary tuberculosis, you can see those shadows. That is a sign of mycobacterial infection. So, human, as I said, these are the main source of the infection. Those that have open cases and they excrete tubercle bacilli from the respiratory tract by droplets nuclei. So they are inhaled and reach the alveoli. So in the host, mycobacteria brought through lymphatic channels, bloodstream, via bronchi and gastrointestinal, mainly by means of macrophages. According to the timing of the infection, we may distinguish primary infection or primary tuberculosis and secondary infection or reactivation of tuberculosis. And you can see here that once ingested or once inhaled, they reach alveoli, multiply there, engulfed by the dendritic cells and alveolar macrophages, and also migrate to lymph nodes. And in the lymph nodes, they activate T and also B cells, but mainly with the T cell activation. So the Th1 cells will be activated to cause granuloma formation. So because of the production of the cytokine stimuli for activated macrophages and other cells. Interestingly, that B cells also will be activated and they finally produce antibodies. But antibodies won't help. They just circulate within the infection but won't help anyhow and cannot make the immunity protective. And this, we may see the granulomas formation, and then when it is emptied, so then mycobacteria can spread through bronchi and follow the dissemination in the body. Usually, what disease is called primary tuberculosis? The one that occurs in the individual that were not infected before. They were free from infection. And it may occur in any part of the lungs. Usually in the base of the lungs. And then mycobacteria spread from initial site through lymphatic to the regional lymph nodes. And the feature of the primary infection are acute exudative lesions develop and spreads to the lymphatic channel and regional lymph nodes. And the involved lymph nodes undergo cassation with usually that usually then calcify. And the hypersensitivity will be developed, and the tuberculin tests start to be positive. But then primary infection may occur in a form of so-called miliary tuberculosis. When the immunity of the body is not so high, when bacilli reach alveoli, they spread directly to the bloodstream, and the bloodstream distributes it over all organs. When focus ruptures into blood vessels, bacilli are disseminated throughout the body, and the numerous granuloma will be formed around the body. Those granuloma have the seeds-like appearance, like millet seeds that we use to feed birds. That's why it is called miliary tuberculosis, because those small yellowish granulomas like seeds can be found in almost all organs. So if cessation lesion discharges its content into bronchi, mycobacteria can be aspirated and distributed to other parts of the lung, or may be swallowed and pass to the gastrointestinal tract and further to the body. So then, what happens after the primary tuberculosis? Usually, we mention that bacteria may survive in the primary lesions. And then those bacteria then overgrow and cause secondary or reactivation of tuberculosis. In the beginning, regional lymph nodes are not commonly involved. They do not cessate. Reactivation tuberculosis usually begins at the apex of the lung because the oxygenation of the lungs is highest there. So reactivation of tuberculosis starts from the apex of the lungs, right? So, but this is not primary tuberculosis. Within the secondary tuberculosis, the chronic tissue lesions usually occur. So the tubercles will be formed, cessation and fibrosis of the granulomas occur. So you can see here that once the droplets are ingested and granulomas are formed, so you can see the structure of granuloma, and you can see that the drained lymph nodes will be infected with the help of T lymphocytes. So how to recognize, the question is it easy to recognize mycobacterial infection? Of course not. First, initially, the symptoms of general inflammation will occur. For example, fatigue, weakness, weight loss, and fever. And if it goes for several weeks up to a month, then it will be maybe the sign of the TB disease. So then chronic cough appear, and first this cough is dry, then the chronic productive cough will appear. Night sweats is very typical, and then low-grade temperature will be developed, and then hemoptosis in advanced state will occur with the blood spitting. So then meninges and kidney can be involved in the process, very common, and meningitis and urinary tract infections can occur without the typical appearance of the typical signs of the lung TB. So, as we said, as we stated, the tuberculosis is the reaction, so is mediated by the reaction of the immune system. So, protective immunity against mycobacteria is the cellular immunity. So, and it is associated with the ability of mononuclear phagocytes to limit the multiplication of the tubercle bacilli. If it happens at the very beginning of the disease, so most probably it won't be produced to miliary, and the prognosis will be more optimistic. The immune response to the bacilli is the cell-mediated type, which is mediated by type 1 T helper that leads to the protective immunity. T helper cells are the major mechanisms. If the immunity is developed against within the initial infection, then it will be associated with the mononuclear phagocytes to limit the multiplication of the tubercle bacilli. And as usually in the immunity, the critical player is the dendritic cell that may activate T helper cell, but also T cytotoxic cells. And also, that is important, T cells that have CD1 for antigen, lipid antigen recognition. As we stated, antibodies will be formed against mycobacteria, but they have no relation to the immune state and the resistance against mycobacteria. So usually, even within the primary infection, the host body will develop hypersensitivity reaction against the components of mycobacteria that result in the Mantoux test positive. My dears, how do we diagnose mycobacteria infection? First, I would like to demonstrate that in the English medium books, you may find, or some advertisement, you may find the new way over the mycobacteria diagnosis, and this is in vitro test. It is called IGRAS, right? So interferon gamma release assay. And that is the whole blood test that can aid in diagnosis mycobacteria infection. How it is performed. So the fresh blood samples are mixed with the antigen. Usually the blood is taken, and we have a stimulation of the blood with the antigen, centrifuge and collect supernatant, and perform quantify gamma interferon ELISA test, and make the calibration standard curve at the beginning with the control samples. Then to see the correlation between extinction found in the spectrophotometer and micrograms of gamma interferon produced when whole blood sample is exposed to the antigen. Because we know that T helper cells in response to the contact with the Mycobacteria antigens start producing gamma interferon. So this is a very new one and I wanted to present it. And this is the oldest one, see? This is the newest one and this is the oldest one. This is in vivo skin test, tuberculin test. So this test was developed to find past or present tuberculosis infection based on this reaction of the T cells inside the body. Tuberculin can be placed into the skin by several ways: injection, scratch, or puncture. Usually we use man-to test, that is scratch. So, and as a result, 72 hours after this test, we may see raised red or hard zone that will be formed around the site of injection. And the person is said to be sensitive to tuberculin because this will be recognized positive. Positive tuberculin test indicates that an individual has been infected in the past and continues to carry viable tubercle bacilli in some tissues. A negative tuberculin reaction does not rule out a diagnosis of active tuberculosis because if we have so-called allergic reaction, it can be inability of the body to respond to the familiar antigen. Usually two types of tuberculins are known. We don't use the old tuberculin right now because this is just a concentrated filtrate of broth where tubercle bacilli had been grown for 6 weeks, and it usually contains tubercular proteins with the different fatty acids. Then most probably we use now so-called PPD, purified protein derivative, obtained by chemical fractioning of the components of tubercle bacilli. So the reaction for tuberculin. So we can see that the test is positive when the induration of 10 mm or more in diameter is developed. So usually induration, edema and erythema developed within 48 to 72 hours. And the tuberculin test become positive within 4 to 6 weeks after infection or injection of the vaccinal strain of Mycobacteria bovis that is called BCG vaccination. Only the elimination of viable tubercle bacilli resulted in reversion of the skin test to negative. But the thing is that negative tuberculin test can be of because of the two different reasons. First, the individual had not any contact with tubercle bacilli yet. Then the Mycobacteria is not familiar antigen for the body. And then the second reason, in the presence of the tuberculosis infection, energy will be developed due to overwhelming tuberculosis or sometimes as a result of previous measles, Hodgkin's disease, sarcoidosis, AIDS, or immunosuppression. Then the body just cannot respond to the typical known stimuli. So, of course, positive tuberculin test does not prove the presence of active tuberculosis. It's not enough. It's just pre-lab initial test. Isolation of tubercle bacilli are needed to prove the presence of active disease due to the Mycobacteria infection. And usually it is done according to the USA, in type 3 biosafety level. Do you remember the previous lecture? And according to the Russian Federation, in type 2 biosafety level. It cannot be done in any microbial labs in the world. For example, not in the lab where teaching occurs. So usually, how do we perform culture? Specimen consists of fresh sputum, pleural fluid, or some other materials. Should be treated first to decontaminate sputum from other microorganisms, and then for concentration of Mycobacteria bacilli. And then as soon as we have decontaminated sample or specimen, then the processed specimen should be examined microscopically, and the same specimen should be placed in culture. So a microscopic examination is quite good, but it is not highly sensitive. So to see them under the microscope, we need to have at least 10,000 to 100,000 mycobacteria in 1 ml. So that's a lot. And that's why the sensitivity of the test is not that high. We may perform Ziehl-Nielsen or fluorescent staining. Then culture is performed when processed specimen is placed most commonly in selective medium. I would like to repeat that antibiotics and disinfectants may be used to make the medium selective. And of course, they grow better in the broth medium. So usually we perform growth within the 8 weeks, and this is the example. And within 8 weeks, most probably we see the growth of bacilli. But for negative result, we need to culture them even longer, from 8 to 12 weeks. Identification of mycobacteria may be used by conventional and non-conventional methods. Conventional methods require 6 to 8 weeks for the identification. They include observation of the rate of growth, because Mycobacterium tuberculosis is slow grower, and we may have some that grow within 7 days. Then typical colony morphology. I think you can see it here. That's how Mycobacteria colonies appear on Lowenstein-Jensen medium. It is believed that they appear as shrink leather. So then pigmentation of the colonies. So what is it, pigmentation? The thing is that all mycobacteria are subdivided into several groups: photochromogen, cotochromogen, and non-chromogen. Photochromogens are those that produce pigmentation of the colony in light but not in darkness. The example is Mycobacterium kansasi. Then scotochromogen that produce pigment when growing in the dark but not in the light. And non-chromogens, which are non-pigmented or have light tan or buff colored colonies, very light. So this is from, for example, avium intracellular complex. The appearance of the colony 3 to 4 weeks after inoculation of this particular type on highly selective Lowenstein-Jensen medium is the way of identification. So it means the most probably we identify them according to the cultural characteristics rather than biochemical tests. The except for that methods of identification, we may use nucleic acid detection. So, for example, we may perform hybridization procedure or polymerase chain reaction. Apart from this, we may perform high-performance lipid chromatography to detect profile of mycolic acids. So then we also may use enzyme immunoassay to detect mycobacteria, antigens or antibodies, but this is not sensitive and it is not usually recommended for routine diagnosis. So, what do we use the nucleic acid probe methods? So, you know that it is quite rapid. It can be performed for in 1 day for identification. And hybridization procedure, DNA probe specific for rRNA sequences of the test organism are used. Double-stranded hybrids are separated from unhybridized single-stranded probes, labeled with chemicals, and activated by hybrid and detected by chemoluminescence. So, most probably now nucleic acid-based techniques are used to perform antibiotic sensitivity testing. So why it is so important? Because Mycobacteria tuberculosis may produce resistance to drugs by means of accumulation of mutations in the chromosome. But then all those mutations are already mapped and we know about them. So it means that the polymerase chain reaction assay mainly now is used not to identify mycobacteria just simply, but to find what kind of resistance this strain of mycobacteria may develop. Because the mutations are mapped for all most common drugs used against Mycobacteria tuberculosis. The way of treatment, my dears, it's a bit different from the others. Because here among the anti-mycobacterial drugs, we clearly develop two lines of drugs. First line drugs and second line drugs. First line drugs, including most effective right now isoniazid, rifampin, and some others. So they are major drugs used to treat tuberculosis right now, and the most strains around are quite sensitive to it. But then if the resistance is developed in the mycobacteria population, then this drug, for example, now a lot of mutations are detected against rifampin. Then this drug will be extracted from the first line and placed into the second line, because mycobacteria in this particular area start to be resistant to it. So usually in second line drugs, we have substances that are a bit more toxic or less effective, or those resistance against which are developed, against what are developed already. So they should be used when first-line drugs treatment failure or multiple drug resistance occur. The risk factors of development of drug resistance in mycobacteria include persons with HIV infection or who are at risk for HIV infection, and also persons who were previously treated with the regimen that did not include rifampin. So, treatment of tuberculosis generally employs not one, not even two quite often, but 3 to 4 different types of drug. And usually the initial prescription is for standard 9-month regimen. But then usually to monitor the possible drug resistance, we need to check the bacterial load in the bodies, in the body of the patient to control the effectiveness of the treatment. That is usually performed with the help of PCR. So, what do we call multiple or multi-drug resistance TB? This is when mycobacteria resistant to both isoniazid and rifampin. So it occurs quite often in certain geographic areas, in certain population like hospitals and prisons, in persons with HIV infection. So persons infected with multi-drug resistant organisms should be treated according to susceptibility test that results from the infection strain. Therapy should include more than 3 drugs, a minimum of 3, to which the organism is susceptible. Prevention and control. Public health control, that is of utmost importance. We have to identify those that may serve as source of infection and treat the patients with this active tuberculosis. But also we have to perform careful follow-up of their contacts with the tuberculin test, X-ray, and treatment. Individual host resistance control. This is the reduction of host resistance by knowing specific factors may convert asymptomatic infection into the disease. These factors are rather social than depend on medical care, health care. These factors that may be really important in activation of the disease are starvation, gastrectomy, with the suppression of cellular immunity. So HIV infection, anti-tumor therapy, and the therapy of the allergy reactions with corticosteroid that inhibit the immunity, and that is really important. Then, to prevent and control infection, we also may perform chemoprophylaxis and immunization, and also try to eradicate the tuberculosis from cattle. Few words about the chemoprophylaxis. It is performed to asymptomatic persons who have converted to a positive tuberculin reaction, and also for tuberculin positive individuals inside a group of high risks of complications at certain age groups, children and teenagers quite often, and persons who must receive immunosuppressive drugs. So then, the question is that, sorry, my dear, sorry. So now we come up to a very serious question, my dears. And the question is whether we have or have no means for immunization. What is the problem that we have with the vaccination against mycobacteria? We know that vaccine exists that is called BCG, Bacillus and Calmette and Variant. And this vaccine contains living a virulent tubercle bacilli. What does it mean? But this is very contradictive vaccine. So it had been developed by Calmette and Guerin within 13 years of adaptation of Mycobacteria bovis. They first tried with Mycobacteria tuberculosis itself, but they failed to develop vaccine out of it. It had been still very toxic. True, they did it for Mycobacteria bovis, and it lost its virulence after prolonged cultivation, but still, Mycobacteria bovis vaccine (BCG) is still recognized as very dangerous in many countries in the world and not necessary for the specific prevention of Mycobacteria tuberculosis infection. There are some countries where BCG is not recommended for routine use of the population, of the whole population. For example, it is not recommended to be used in the United States. There, it is recommended to be used only in the regions where the special ethnic groups are living. For example, in Chinatowns, where Chinese people live, or in the areas where black African Americans, are living. So, but then the thing is that because of lack of the BCG injection, so then the generally in America there are a bit different norms and standards for using men-to test, because the general population has not been exposed to the Mycobacteria tuberculosis. So within the vaccination, so the development of the positive reaction is a bit different in the mechanism. So in our countries, but also in India, I think, in Nigeria, and a lot of other countries, BCG vaccine is one of the very first vaccine given to a newborn babies before they leave the labor house wife or nursery wife. So, because it is considered that vaccination should be given before then the hosts or the organisms come in contact with the environmental mycobacteria that may cause the rising of the tuberculin test. So, and one of the important things is that the only live virulent vaccine may cause cell-mediated immunity that is needed for development of the protective immunity against mycobacteria. So, because it is a live vaccine, it has a very long list of contraindications. It should not be given to pregnant women. It should not be given to immunocompromised individuals, and also to individuals with a long list of the diseases. So, still, there are speculation whether we can replace BCG vaccine with something else or not. Several candidates are developed with the recombinant polypeptides or proteins of Mycobacteria tuberculosis together with some lipids. But then the effectiveness of these candidate vaccines are not proved yet. So another direction in which the research work is going on regarding the use of BCG vaccine is to improve the way of the delivery of the antigen into the body. Usually it is given subcutaneously, but also, for example, in Japan, the special delivery instrument is developed. You can see it here, by the way, with the 7 needles that are soaked in the vaccine here and injected intracutaneously into the individual's forearm. So, still, BCG vaccine is given to children in many different countries. So, in a lot of countries, it is considered to be quite effective. And this is also considered in the Russian Federation. But the thing should be taken into consideration that the immunity against mycobacteria are considered non-sterile. What does it mean? It means that the immunity may be developed within the time when mycobacteria, alive mycobacteria, are still may reside in the body. So if you, my dears, take a look at your left, I think, forearm, you can see a scar. That is a result of BCG vaccination. It is believed that to develop immunity against Mycobacteria tuberculosis, we should cause a quite strong inflammatory response, local inflammatory response in the primary site. And it is believed that if the scar is formed on the site of mycobacteria injection, then the immunity is developed properly. If there is no signs of vaccination, possibly the injection of the vaccine was not, or vaccination was not sufficient to develop immunity. And as I said already, the individual may stay man-2 positive for quite a long while after vaccination. For example, for a year or so since the one was vaccinated. My dears, we work without the break, so this I think considered is enough, so we may finish with this. And please take a look at the books also and be ready to discuss mycobacteria during the classes. With this we finish. Goodbye, my dears. The lecture is over.